So we'll go on to our fourth and last speaker for today, who is Dr. Dan Geschwind from UCLA. I'm sure he's here. Dr. Geschwind is the McDonald Distinguished Professor in Human Genetics, Neurology and Psychiatry at UCLA. He is a Senior Associate Dean there and an Associate Vice Chancellor for Precision Health, and he leads the Institute for Precision Health at UCLA. His laboratory has really pioneered the application of systems biology methods to neurological and psychiatric diseases. He's put considerable effort into fostering large-scale collaborative patient resources for genetic research and data sharing in autism research. He serves on many national advisory boards. He's won many prizes over the last number of years and published over 400 papers. He was elected to the American Association of Physicians and the National Academy of Medicine. Dr. Geschwind is going to summarize work showing how to leverage genetic findings and genomics to understanding neuropsychiatric disease pathophysiology. Dr. Geschwind.
Yeah, so I'm at UCLA, not at McLean Hospital, just to clarify that. I hope that's clear. But, and that's why it's still about, something like 10 of 8 in the morning for me. So I thought I would give a brief and kind of somewhat, you could say superficial, but look at how we think about genetics and how we need to move from finding genes to understanding their function. So hopefully it will become clear. These are my conflicts of interest in terms of advisory boards. Now what I'm going to describe is what we think of as a simple paradigm. And I want to illustrate one thing here. It might be a little hard to see, but essentially what is shown there in A, B, and C are high prevalence of all the major psychiatric disorders ranging from schizophrenia and autism at the bottom and Tourette's syndrome all the way up to anxiety disorders and major depression. All of you know that. But the other, B and C just shows the high and kind of consistent heritability in all of these conditions. And so, and C shows the SNP heritability, or the percentage of heritability that currently can be measured and attributed to common single nucleotide polymorphisms. It's probably an underestimate for sure. But so the simple paradigm then is, because these disorders have a large heritable component, is to find loci, that is, do genome-wide studies that identify regions in the genome that increase risk for these disorders, and then find the genes, because most of the loci are actually outside of coding regions. Only about 3% of the genome is protein coding, and so it's not surprising that most of the loci lie outside of those regions. So one has to, and as it turns out, we're finding a lot of loci, we're finding the heritability, but actually attaching the functional implication of those loci to genes is actually quite tricky. Even more tricky is this last very vexing part, determine how they cause disease. And at some level, I'd have to say, when I started about 17 or 18 years ago thinking about autism, I thought maybe we'll find a few genes in my entire career, and one could say that we have been blessed by extraordinary success across psychiatric genetics, largely because of large-scale collaborative projects that have enabled one to gain the sample size and power to identify, genes. But they do provide a critical functional anchor, but determining how they cause disease is not so easy. And then another point I want to bring here, on the y-axis it says genetic correlation if you can't read it, and then on the bottom are the different disease pairs. And the point here is that these genetic correlations are non-zero. Maybe not surprising for bipolar and schizophrenia, but a 0.2 genetic correlation between autism and schizophrenia shows that at a genome-wide level there's a really quite significant, although it's not the majority of loci, but quite significant overlap. So one of the challenges we have is to understand disease specificity, what are common risk factors, what are shared, and how do these load on to brain mechanisms. And you just heard a very elegant talk that's been going at one of those very common disorders for quite a long time. So I'm going to focus on autism, which has been a major component of my lab over the last 15 years. And so one point I want to leave you with that I'm sure you actually probably know already is many forms of genetic variation and modes of inheritance. And this is meant to, on the left, is just really from 2 years ago, a bunch of the rare genetic variants and syndromes that cause, that are major forms of autism, most of these being syndromic. But many of them are newly found with whole exome sequencing studies that have been done. And the point is that none of these are really common. Even the most common of these rare disorders is less than 1%. If you add all these up and collapse them in 2016, they accounted for about 3 or 4% of autism cases, and it was projected that maybe it will account for about a fifth or so, plus or minus. So rare genetic variation, especially de novo variants, which are non-heritable, like a Down syndrome, actually account for about a fifth of autism. But the heritable component has not been that well deciphered yet, and there's quite a bit of work going on there. So what it shows on the right is there's still a lot of gene finding to be done. But even at this point, we know that there are going to be many genes. None account for more than 1% of cases. They're very highly additive effects, that common genetic variation does play a substantial role. And there's strong pleiotropy in that many of these loci, especially the major loci, give risk for not just autism, but autism plus intellectual disability, various epileptic encephalopathies, and even schizophrenia with intellectual disability. So again, as I said, we're kind of, every few months now in autism, there are another dozen genes or so being identified, which is very exciting. And these give us really critical clues and hooks to begin to develop therapeutics. But it does lead to also several challenges that should be obvious, hopefully, that there is huge genetic heterogeneity at the population and individual level, but also genes don't act in isolation. They're components of biological signaling pathways and in the brain complex neural networks. So one of the questions we've been asking is, can we identify patterns within these large dimensional data sets, such as the genome or the transcriptome, that are related to key aspects of biological function or disease pathophysiology? And then can we use these patterns to inform treatment development? And the idea is that rather than treating these as unidimensional single levels of analysis, we're trying to integrate all the way at the bottom from, let's say, a disease phenotype or clinical measurements, behavior, et cetera, all the way up from sequence data to all the way up through transcriptomes to these clinical phenotypes. And if we can kind of do this in a coordinated fashion rather than one at a time, we're likely to have a lot more power, this kind of systems biology approach. And we're moving in that direction, and we're just on the threshold of having the large enough data sets to begin to really integrate in this manner, which is very exciting. So the way that we've kind of thought about this is, of course, you have genetic variation that works through all of these different stages, DNA to RNA to protein. But in the nervous system, we have this other complexity, which is synapses, circuits, and behaviors. And connecting this is really a daunting task, but it's something that we are faced with and must address. So one way that we've thought about addressing this is in networks. We know that both the brain and genes act in networks, and I'll describe that in a second. The focus of our work, and what I'm going to talk about today, has been at the mRNA level, which I have there in red. And the issue is not that mRNA is the be-all and end-all, but that one can measure it at high resolution, pretty much from the lowest abundance to the highest abundance, so very high dynamic range, in many samples very inexpensively. And as we can do that with protein and these other levels, these high resolution and high throughput measurements are going to become really critical. But because we can do it in such high throughput, we focus on the mRNA. So again, genes don't act independently, they form networks. And what I've shown here are 3 groups of genes that go up and down in kind of shared patterns as groups or clusters or what we call modules. And one can identify genes that kind of go up and down together across samples in multiple conditions and identify them as modules. And we and other people have shown that these correspond to very elemental aspects of neurobiological function. So for example, shown all the way on the right is if you took whole brain data from hundreds of microarrays from different laboratories and asked and identified these covariant structures, identified specific modules, most of them correspond to the main source of variance in the tissue, which is not surprisingly the cell type abundance, so different proportions of different kinds of neurons, glia, et cetera. So you can do an in silico dissection. And just to give you a kind of more formal look at this, usually when people do gene expression, they're looking for genes that are up and down, and that is fair. You want, and it's often reasonable to focus on the gene that is the most down or upregulated in your condition. But usually what that means is people are looking under the street lamp. And here I've shown two genes that are not differentially expressed in the experimental and control situation. However, if you look at their covariant structure in terms of their correlation, one can see that in the control group, these genes are highly correlated, and the experiment they're not. So something's happened about the relationship of these two genes to each other between these. And essentially that's what we're measuring, these relationships, these co-expression relationships, rather than just, is the gene differentially expressed? And actually this takes advantage of all the kinds of variance that's in your experimental data and can really lead to much increased power in gene expression experiments. And one of the kind of mathematical kind of, graph theoretical formalisms that really makes this even more powerful is the fact that gene expression obeys the kind of maps and graphs that we see on the right, the airline map. And intuitively, hopefully you can understand this, so that these are scale-free networks. A small number of nodes have a large number of connections. Most nodes have very few connections, and the few highly connected nodes are essential for the integrity of the network. In other words, when there were storms over Newark yesterday, and I was coming in from California, kind of everything was delayed at LAX, even though it had nothing to do with what's going on at LAX. So the point is these nodes, these hubs, really are key drivers and markers for the main biology. So one of the things this allows us to do is to take the genome, summarize it into a set of modules of co-expressed genes, going from tens of thousands of genes to dozens of modules, and within those modules, dozens of driving hub genes. So now what I'm going to talk about is how we kind of take this approach and determine how genes might cause disease. And of course, I say this in quotation marks because all of this work, nobody really knows how these genes really cause disease yet, but I'm going to summarize our progress. First, I'll talk about convergence in postmortem brain, and second, I'll describe the overlap with other psychiatric disorders, which is a really critical issue. So when we're looking, I just want to emphasize at postmortem brain, we can't really, just from gene expression alone, if we find changes that are up or down or modules that are seen in autism versus controls, we can't a priori ascribe causality to that. That is a molecular pathology of the disorder, but we don't know that it's causal. It would be like seeing amyloid in the Alzheimer's brain without knowing that there are amyloid mutations. You don't know that the pathology is causal, you just know it's associated with disease. But what's really striking to me is that although at a gross macroscopic level, there's no neuropathology in most of these disorders, there is at the molecular level, and we've been doing a lot of work to define that. So we reasoned almost a decade ago that despite this etiologic heterogeneity that was becoming obvious, is there a shared pattern of molecular pathology in postmortem brain from subjects who had autism? So what we found, to summarize, is 2/3 of cases show upregulation of a module that is really represents the normal microglia and astrocyte modules seen in normal brain. There's a parallel downregulation of neuronal genes involved in vesicle transport and synaptic signaling, and that the causal genes predominantly affect the neuronal component. In other words, if we ask, where do the heritable components partition, where do mutations lie, they lie in the neuronal, not in the glial components in autism. And what I've shown at the bottom there is just some replication using RNA sequencing in a much larger sample that shows further refinement of these patterns. So we have multiple neuronal downregulated modules and a separation of the upregulated astrocyte and microglia modules in autism in the more recent paper. Furthermore, it shows in a monogenic form of autism called 15q duplication, which is often inherited from the mother. Those patients share this pattern, even in a more homogeneous way than patients with idiopathic autism, and that this pattern extends beyond coding genes to microRNA and long non-coding RNA as well as splicing. And just to kind of summarize that very briefly in this short talk, is that when we look at microRNA, we can also see that there's a microRNA pattern that largely separates about 2/3 of the cases from controls, shown in red there. And when we look at H3K27 acetylation with Shyam Prabhakar in Singapore, H3K27 marks active enhancers. We can see that there's a strong relationship between the acetylation of those enhancers and changes in gene expression. So one of the key points here is that multiple methods, we're not just relying on RNA sequencing, which has many confounders, but that multiple techniques support the fact that there is a shared molecular neuropathology in at least 2/3 to 3/4 of patients with this disorder, which is really quite remarkable and to me was unexpected at the time. So then another major thing we were able to look at in the more recent study was trajectory. And so we had many cases below age 15 in this more recent study in 2016, and we were able to see that the microglia module did show a temporal trajectory that was very unusual. The black is the control, the red is the autism, and what you see is that normally over the first decade of life, microglia activation and genes in microglia are substantially downregulated. And so the upregulation that we're seeing in autism is more like a failure of the normal developmental downregulation during development. And again, just one idea is that we don't know whether this is adaptive or maladaptive, but it does provide a potential treatment window during the first decade of life. So how does this make me think about the molecular neuropathology of autism? And I think about this coming from a medical perspective. We think in general, in big terms, cancer is a disorder of proliferation and invasion, asthma is a disease of hyperreactive airways, stroke is a disorder of acute brain hypooxygenation. All of these have a number of different molecular, genetic, and environmental causes. In this context, what is autism? And so using the molecular neuropathology as a guide, we can say we have altered neuronal gene expression and increased microglia and astrocytes. The altered neuronal gene expression is predicted to cause synaptic dysfunction. And again, we don't know whether the astrocyte or microglia is adaptive or maladaptive, although we probably, having this chronic upregulation, we would presume or guess is probably maladaptive. There's also a really fundamental role becoming more and more apparent for alternative splicing and alternative isoform usage. But all the genetic variation that we're able to capture at this point sits in the neuronal. In other words, that's where the causal arrow sits. So this is our current kind of very, you could say it's a vague model, but it's a starting point for beginning to understand what may be going on in the brains. And now the real challenge is to go all the way back to the initial genetic insults to understand how a genetic insult might result in this. And so as we've been thinking about this, we also asked, and this was led by Mike Gandel, who is a MD, PhD psychiatrist and now has his own lab at UCLA, how do these changes in autism relate to other psychiatric disorders? So the idea is you have multiple causes, multiple etiologies of these disorders, but one place of convergence. Think of it as a quantitative phenotype, similar to imaging, except for this is at a very, very refined molecular level, and we can do it quite quantitatively. And again, the point being that I showed earlier, this is from a PGC paper earlier in Nature Genetics, that there is substantial shared heritability across these disorders. And so Mike, painstakingly, over about 18 months, was able to download and normalize and process almost 800 microarrays that had been done mostly by other laboratories. These are published data in other disorders. The difficulty was getting them into kind of a single framework. And again, I just want to make this push for data sharing. Without having this available, the study would never have been doable. And so just to summarize, what we see is a very striking set of correlations. Autism is about 0.4 correlated with bipolar, more so with schizophrenia. What's interesting is that bipolar and schizophrenia are far more correlated with each other than, let's say, bipolar is with depression. And some people have taken offense to this, saying, well, bipolar is a mood disorder, why isn't it closer to depression? And I can't tell you, I can just tell you that these are the data, and these are telling us something about these disorders that we should really pay attention to. The genetic correlations would tell you the same thing. And so autism also has a very slight, non-significant correlation with inflammatory bowel disease that turns out to be the cytokine immune upregulation component, which in the bowel, in the gut, is due to different cells secreting some of these same cytokines versus microglia in the CNS. Another thing we see is that alcoholism really does not connect to any of these diseases at the global transcriptome level. Now, what's interesting here is now, what's causing this? And so most of the reviewers said, well, this is just postmortem artifact. Well, here's one piece of evidence. There's a lot of evidence that it's not an artifact. We took the pairwise correlation in transcriptomes and asked, is that related to the SNP-based correlation from genetic studies? And the answer was shockingly huge. Almost 50% in the variance in the transcriptome overlap can be explained by shared genetics. And I can tell you the shared genetic components that we now know, except for maybe for schizophrenia, are quite underpowered in these other disorders. And so this is likely an underestimate. And again, you can see autism and IBD, basically zero shared genetic component, again, consistent with the fact that what we're seeing there is really a convergence at the level of shared mechanisms in neuroinflammation and gut inflammation. So now we can use the networks to begin to parse out the biology. We show global transcriptome overlaps, but now we want to say what aspects of this biology are specific. And so, not going to spend too much time on this. It just basically shows that the modules, we can identify a bunch of modules, and they cluster into cell types. Neuronal modules at the upper right, glial at the bottom left. And so I'm just showing you an example here from the paper that shows how these modules go. This is the first principal component of gene expression within these modules. The first four are neuronal modules. The yellow is the astrocyte, and the lime green is microglia. And what one can see is that astrocytes are upregulated in schizophrenia and autism, but not microglia. In fact, autism has a very specific large-scale upregulation of microglia that is not observed in the other disorders, and that we've now been able to confirm with extensive RNA sequencing. So one of the questions is, where does the genetic variation lie? And again, just similar to the autism study, the genetic variation here lies in a few of these neuronal modules are substantially enriched for GWAS signals as well as rare genetic variation. So you can say, okay, so you know these pathways, where does that bring you? Well, you can begin to now ask, what biological features, what kind of neurophysiology, what kind of cell-cell interactions can lead to these types of transcriptome alterations that we see? At the mesoscale, one can connect gene expression to resting state networks. And this was a study that we did with Jenna Konopka at UT Southwestern, replicated in multiple data sets. I just want to bring that up, that all the data I'm showing you is always replicated in more than one data set. And what we see here is that default mode activity correlated genes are enriched in the modules that are downregulated in ASD brain. And at the microscale, we had done a previous study published in Molecular Systems Biology where we had identified modules that corresponded to neuronal firing rate. And again, one of these, this purple or magenta module, was substantially downregulated, both in autism, schizophrenia, and bipolar. So again, showing that even cellular studies in a dish or studies connecting brain imaging to microarray data from postmortem brain can begin to give you clues as to what some of the underlying biological processes are that are driving these things. So certainly activity-dependent gene regulation and splicing is at play here. So what we're looking at in the postmortem brain is not the direct effect of a genetic perturbation, but the effect after multiple stages of neurodevelopment and neurogenesis and behavior of activity-dependent changes in the circuit that are common across patients. So again, I told you it's difficult to pick, to find causality, but we can begin to go from the other angle and say, well, now that we're identifying all these autism risk genes, when and where are they expressed? And so we and a number of other people have done these studies where we've taken data and asked what cells do they act in and what epochs in development. And so just to summarize this, we basically take, shown by the arrows, large effect size mutations, small arrows or small effect size variants, common variants. And we can take these different classes of genetic risk factors and ask, if we look at normal brain development from fetal life to 3, 4, 5 years of age or adult, do these genes cluster in particular biological processes or modules or epochs during development? And to make a long story short, the large effect size mutations cluster in chromatin modifying genes that are involved in transcriptional regulation during cortical neurogenesis and probably during neurogenesis of other brain structures. And so to summarize what has been found in this kind of graphic, if you look at rare de novo large effect mutations at the top versus genes that are differentially expressed in postmortem brain versus candidate genes with lower effect sizes, and compare that to genes that cause various forms of intellectual disability, Mendelian forms, one can see in the second panel that they fall in different molecular networks, that the red de novo genes fall into genes that are expressed early prior to birth, and that the other genes, those that are differentially expressed in lower impact candidate genes, are expressed prenatally, but they're expressed after neurogenesis and continue to increase subsequent to birth. So ASD genes from multiple sources converge on prenatal neurodevelopmental programs, but they're distinct depending on the effect size. Transcriptional and translational co-regulation actually link these at multiple levels, and that's data I haven't shown you that's in this paper in Cell in 2013. But one of the things that we found, and Matt State's group found as well, is that these risk genes, especially the de novo ones, are enriched in glutamatergic projection neurons, and that these patterns highlight features that distinguish autism from intellectual disability, because the intellectual disability genes, as writ large, didn't show the amount of enrichment in any of these pathways. So this really begins to give us a molecular architecture of autism and to begin to be able to think about, kind of what's happening at a molecular genetic standpoint. On the left, I'm showing, essentially natural selection. Red are large effect major mutations, like Down syndrome, like CHD8 mutations, like 15q duplication, etc. And what you can see in the population history there is that red genes don't go that far back in history. Blue genes do. Those are things with very small effect sizes. It's because natural selection has weeded out things with large effect sizes on fecundity, and that those things are going to really mostly be de novo or very recent mutations. So again, what we're finding in autism is that those affect transcriptional regulation during neurogenesis, and it fits with a molecular and transcriptional hierarchy where you have a 3-tiered level of regulators, and that these early acting, really catastrophic genes are top-tier regulators, not surprisingly, and the kind of risk variants are occurring in synaptic development and function for the most part, and those are the genes that are regulated by these top-tier regulators. So what this tells us, again, is that the genetic architecture parallels a molecular regulatory architecture. But it also tells us we have an enormous knowledge gap. We need refined understanding of these regulatory networks during neural cell lineage specification and development in humans. That is an enormous gap, and these various efforts that I cite there, PsycEncode, the Brain Single Cell Initiative, etc., are trying to fill that in. Once we have that, we'll have a much more refined map about when and where these genes are acting and what processes they're actually disrupting. And so part of our work has been to help build this reference map. We've been working in fetal cortex. And so we've asked 4 basic questions. What genes are expressed? There we use gene expression. What regions of the genome are active? We look at chromatin accessibility using ATAC-seq. What genes do they regulate? We look at 3-dimensional chromatin interactions. As it turns out, most regulatory elements in humans don't act on the closest gene. More than half act on something distal. And the chromatin is folded in a 3-dimensional structure where we can actually elucidate those regulatory relationships. And then how do they regulate them? What transcription factor binding sites are in those enhancer and promoter regions? So we separated the brain into the germinal zones below and the developing cortical plate, and did a number of these studies, both the high-C study of chromatin structure published in 2016, and more recently an integrative study in 2018. And I just want to share a couple of, of what this really shows us. So again, gene regulation occurs via chromosome folding that brings regulatory regions into contact with their target genes in a tissue-specific manner. And what we can do is identify these gene loops at 10 kb resolution throughout the genome using high-C, and we find that only half the GWAS loci are acting on the closest gene, which is very consistent with EQTL data that's been published as well. Many are acting more than several 100,000 base pairs away, and we can validate these in vitro using CRISPR engineering. And so this is just, again, only half are acting at the closest gene. This is just showing one of those loci. And dozens of new risk genes are identified, and this implicates pathways that would not have been identified in the GWAS if you just use linkage disequilibrium. And as it turns out, linkage disequilibrium really has nothing to do with these functional relationships. It has to do with ancestry, not with genome function. So we identify pathways involved in schizophrenia implicating cortical neurogenesis and ACH receptor activity, which had been implicated before with a lot of functional studies in humans. So another thing we can do is look, and we can begin to partition genetic signals from depression GWAS, schizophrenia GWAS, ADHD, et cetera, across, and you see some of this here. And then we can ask, are these non-coding hits in regions that are active in fetal brain or adult brain? We can begin to go to a cell type specific level. And so here what we see is that even some adult disorders, let's call it schizophrenia, which appears in, we've known for a while it has a neurodevelopmental origin, and this is just more evidence of that, but they're both, it's partitioned between fetal and fetal and adult brain peaks. Educational attainment, more so in fetal peaks, et cetera, ADHD, fetal. What's super interesting though, if we partition the fetal into the neurogenic zone, that would be on the left, the GZ greater than CP, or the non-neurogenic zone, those things in the postmitotic neurons, we can see that most of the signal is coming for the GZ greater than CP. So again, most of these signals are occurring and having a large effect, and it could be an effect size issue in early neurogenesis, which again speaks to the importance of the fetal life in risk for these disorders. But it also tells us that we can begin to study these in a dish, and so we can begin to use iPSC-derived neurons, and these, you heard a beautiful talk earlier in the morning on using 3D organoids, which many labs are now using. And one thing we can show using the gene networks is that indeed these autism risk genes are co-expressed in vitro in these systems in a manner similar to their in vivo. So it tells us that we can use these, that they're very, very valid models for studying the effects of these developmental genes. And I just cite some of the papers here. But another thing that's super interesting that I glossed over when I showed you the transcriptome patterns is many of you were wondering, what's the effect of drugs? Are those schizophrenia and autism overlap? Is it due to neuroleptics or other drugs? No, as a matter of fact, neuroleptics, common neuroleptics, as we show in a supplemental figure in that paper, Gandel et al., actually push the genes towards normalization. They're the opposite. So they're kind of, treating the pattern in a way. And genes that are drugs that are propsychotic, like PCP, actually partially recapitulate these neuronal patterns. So we've done other experiments in other disorders, but the idea is maybe these transcriptome patterns can serve as a way to screen for drugs. The idea is that we take expression patterns, and there's something called the Broad Connectivity Map that is just one of these resources, where there's 7,000 and now more expression profiles in cells after treatment with FDA-approved compounds. Well, you look at your gene expression in disease model or in patient tissue, and you do pattern matching to find a drug that is predicted to reverse that. And then you test the drugs in a model system. The difficulty with autism and schizophrenia right now is that we can't be sure exactly what the physiologic phenotype we're looking for is. And so it makes it a little bit difficult to do the secondary screens, but I think we're moving there quite quickly as people study the physiologic effect of these genes. So I'm going to end now. I think there's a lot of hope. Network approaches provide an unbiased systems-level framework for integrating multi-omic data, gene co-expression networks and postmortem brain from patients with autism, and those representing human brain development normally indicate points of convergence in autism using very different methods and approaches. And transcriptomic networks illustrate a clear pattern of shared and distinct biological molecular pathology across these major psychiatric conditions, and they provide an initial reference point for understanding the role of genetic risk. And of course, as I mentioned, prenatal cortical development is a critical kind of flashing sign for us. And I hope that I've left you with the thought that bioinformatic analyses, coupled with experiments of large data sets, permit strong hypothesis generation and can serve as a basis for drug screening in complex conditions. So thank you. Well, I was going to thank everybody. Can I have the last slide? I need to thank everybody in the lab and the pictures.
While you're thanking everybody, Dan, maybe anybody with a question can walk to the front to walk to a microscope.
Okay, everybody go eat or take a break, but I really need to thank all my collaborators, the PsychEncode Project, BrainSpan, and our funding sources. Thank you very much. Again.
If anybody has any questions for Dan, please come up to the microscope. If nobody has any additional questions, please join me in thanking Dan Geschwind.